Bonsoir, good evening, everyone. Welcome to the last MEG seminar of the year 2020. Okay, I promise you all, 2021 is going to be much better year than 2020. But in terms of seminars, this has been an amazing year. So we are celebrating a year of this project. So all the way through up and downs of 2020. The students of the Magistère have been hosting speakers from across the world. This is the last one, 2020. We're over 60 participants. So bravo to all the team and to Madeleine for organizing this. Please come back in 2021 because we're already lining up speakers, including joint sessions with students in other cities, in other countries. So, and if you want to be involved, just contact Madeleine and get involved with the team. So Joanne is gonna be host introduce the speaker and host the questions. We have a little bit of technical problem, but we'll manage. And just a last shameless plug for the Lonely Pipette podcast. So we just launched the 6th episode today with Marie Elena Torres Padilla, who gives a fantastic description of her journey from Mexico through Paris to London, to Cambridge, and then back to be a director of a unit in Germany. Please listen, it's there for you, and please subscribe so that we can send you emails and keep up with you. Wishing all of you a fantastic end of 2020. And a much better 2021. And thanks to all the students for in difficult situations for showing so much enthusiasm just for the science. Joanna.
Thank you, Jonathan. So I wanted to thank you all for coming today. We are over 60 participants and it's amazing to see you all, and I hope everyone is okay. Just a few quick reminders. So how we proceed in those seminar series, maybe it's different where you like webinars or the webinars you attend. But here we ask you to have your mics and cameras off. And we will ask you to leave the questions you may have in the chat, and I will read them out loud to our speaker. So today's edition is in the honor on the theme of the Téléthon. And we would like really quickly to remind you that they have a TV program on Friday, and you can donate for the Téléthon on the website don.téléthon.fr. And we are hosting the Student Association of the BGA, who is the Student Association of the Magistère, is hosting a lot of events to collect funds to give them to the Téléthon. And so the Téléthon, the AFM Téléthon, is an organization that helps patients with rare genetic diseases, and it's giving a lot of funding. It helps to fund the institute our guest works out. So our guest is Karim Ben M'Barek. He's a researcher scientist at the ISTEM Institute that works with the Téléthon. So he has a PhD that he was working on in Institut Curie in Orsay, and he worked on the implication of certain proteins in Huntington disease. And then he did a first postdoc at the Institut de la Vision in collaboration with ISTEM. And it was on retinal bioengineered tissues on rodents and primates, non-human primates. And a second postdoc at the ISTEM this time on tissues, on clinical applications for tissues that were formed from specialized cells derived from human embryonic stem cells. And now he's a research scientist. And he works on the development of cell-based therapeutics alternatives to treat retinal degenerative disorders, and strategies based on bioengineering with human pluripotent cells as cell sources. And today he will present us a talk about development of a pluripotent stem cell therapy for the treatment of retinitis pigmentosa. So it's a pleasure to welcome him, and let's hear what he has to say.
Sure, Joanne. So my talk today will be focusing on a specific application of cell-based therapy using human embryonic pluripotent stem cells. And basically, you will see how we started with preclinical evaluation of such therapy in rodent modeling retinal diseases to the translational perspective, and then to the first clinical trial for this application in France. So as Joanne told you, I'm working in EUR, which is an institute dedicated to the study of pluripotent stem cells in specific applications related to monogenic disorders. And basically, we use pluripotent stem cells from patients or without mutations, and we try to decipher the molecular mechanisms that lead to abnormalities in cells who could be reproduced, who could reproduce human diseases. So some basic elements regarding the retina. As you can see here, you have the structure of the human eye, and basically what you have here in red is the retina. And more importantly, this retina is involved in the transmission of light signal into electrical signals, which are then transferred to the brain. And basically, when you look more precisely into this retina, you have a specific structure called the macula with a foveal pit. And this structure is essential for the vision, the precise vision. Indeed, this structure is essential when you need to read or to drive or to have precise movement. When you look into detail into this retina, here you have the different retinal layers. And basically, this retina is composed of neuronal cells. You have first photoreceptors who transmit the light signal into an electrical signal, which is then processed by different neurons in the deep layers of the retina, which are then transmitted to ganglion cells. And the axons of ganglion cells constitute the optic nerve, who is connected to other brain structures. And what is interesting is that this retina is associated to a small epithelium, a monolayer epithelium, which is called retinal pigment epithelium. And this epithelium is involved in the homeostasis of photoreceptors. And basically, when you have a defect into this retinal pigment epithelium, you will have the progressive death of photoreceptor cells, which lead to defects in vision. When you look into this retinal pigment epithelium, they have this epithelium that we name RP, has a number of functions who are, as I told you, essential for the homeostasis of the photoreceptors. And basically, this epithelium is connected, makes the interface between the retina and the choriocapillaris. And this choriocapillaris, through this retinal pigment epithelium, you have an epithelial transport of glucose and vitamin A, and also the transport of other elements, which is really essential to provide energy to photoreceptors. This epithelium is pigmented, as I told you, and this pigment is also involved in the light absorption in order to, when you have the transmission of light signals, they will activate photoreceptor signals, and then they could diffuse inside the retina. Therefore, when you have the RP cells which are pigmented, they will collect this signaling, which limits the reflection of light. This epithelium is also involved in the visual cycle of specific elements, which are essential also for the vision of humans and rodents also. And finally, this epithelium is involved in the secretion of different trophic factors. In the apical side, for example, you have the release of PDF, and in the basal side, you have the release of VEGF. This epithelium is affected by several pathological conditions. You have mainly two conditions in which this epithelium is affected. The first one is a monogenic disorder, which is called retinitis pigmentosa. And basically, you have several mutations that could affect both photoreceptors and RPE, or specifically photoreceptors or specifically RPE. And this group of monogenic disorders is composed of many point mutation in several genes, but each mutation in one gene can cause retinitis pigmentosa. And when you focus on genes who cause an RPE dysfunction, you have 5% of all retinitis pigmentosa who is caused by an RPE defect. The other main conditions, which is more frequent, is age-related macular degeneration. This disease is multifactorial and polygenic, which affects millions of patients in France. And the perspective worldwide is comprising several hundred of millions of patients with all stages of AMD. And when you are focusing on treatments, you have almost no treatment for retinitis pigmentosa. Recently, there was a gene therapy for RPE65 mutations, but it concerns really a minority of patients. Regarding age macular degeneration, you have several forms, the dry form or the wet form, and there is a treatment for a small part of this disease. And globally, what we can say is that there is a global unmet medical need for the vast majority of patients. In AMD and retinitis pigmentosa, you have so this defect in RPE cells, and basically the main strategy of cell therapy is to replace dead cells or deficient cells by new ones. So what I'm going to present you is first proof of concept realized in preclinical animals models, and from all this data that we learned in vivo and in vitro, we developed a clinical trial based on this strategy. So I will detail what type of cells we used, how we formulate the cell therapy in order to deliver it into animals models first, and then into patients, and how we moved from preclinical data to have a clinically compatible cell therapy with all the hurdles associated with this kind of development. And finally, I will give you some perspective toward an industrialization to treat a large number of patients. So first of all, our cell source is human embryonic stem cells. I'm sure that you know how we can obtain human embryonic stem cells, but I will give you just few elements. Basically, these embryonic stem cells are derived from an embryo at a blastocystic stage, and basically we collect the inner cell mass, which is this region here, and we can derive it into culture, and we generate lines of human embryonic stem cells. We have another source of pluripotent stem cells, which is called induced pluripotent stem cells, and these cells could be derived from any adult cell type of an adult body. Here you have the example of patient biopsy. And basically, we can use a cocktail of pluripotency factors in order to reprogram these adult cell types. And basically, we obtain these induced pluripotent stem cells which have the same characteristic of embryonic stem cells, the same characteristics without these ethical issues associated with the use of embryos. And this induced pluripotent stem cells is quite the discovery of induced pluripotent stem cells is quite recent, as you can see, and mainly most of the clinical trials who were done worldwide were based mostly of embryonic stem cells. And since 2018, now there is a switch to human-induced pluripotent stem cells, which most of the clinical trials starting with cell therapy are using induced pluripotent stem cells. So in our case, we use human embryonic stem cells, and basically what is important with pluripotent stem cells in general, including embryonic and induced pluripotent stem cells, is that they hold two essential capacities. The first one is that you can amplify these cells for virtually an unlimited number of times, so you can generate large bank of cells. And the other point is that these pluripotent stem cells could give rise to any cell type of the human adult body, meaning that with these cells, you can generate neurons, you can generate blood cells, you can generate retinal cells in our context. You just need to define a specific protocol to guide these pluripotent stem cells, to guide them through the development in order to obtain the cell type of interest. And with this kind of cell source, as I told you, you can have an amplification potential that allow you to treat millions of potential patients. And you can develop protocols to differentiate these pluripotent stem cells into RPE cells. And you can generate one or several master banks, and these banks are homogeneous between different batches. And most importantly, they are suitable for an industrialization process. This industrialization process is essential to reduce the cost of cell therapies. So major efforts are performed worldwide to use this kind of cell source. So just a few elements regarding the manufacturing process is that we work with frozen cell banks. We have a human embryonic stem cell bank that is frozen, and we develop a protocol. We use a few vials of these cell banks in order to develop our differentiation process, and we generate banks of human embryonic stem cells derived RPE cells that we also froze as cell banks. And basically, with these cell banks, when you need to treat a patient, I will show you how we do, but basically we have an homogeneous cell bank. Each vial in this cell banks is identical to the other. And once we have this kind of frozen cell banks, we have to perform a number of quality control in order to determine that these banks is compatible with the clinical application. And basically, we have some documentation that you need to have to ensure that your cell bank is conform in terms of donation, in particular of human material. You need the informed consent, for example, the patient screen for classical human viruses, and also an anonymous processing. In case of RPE bank, you need to define the identity. Basically, we use RPE cell markers by PCR, by IF, and so on. For the human embryonic cell bank, we determine its pluripotency using also markers of pluripotency and so on. We need also to characterize the purity of the cell. Basically, you need to have a traceability of your cell identity all along the process. So at the beginning of the process, you have a cell line who was characterized in terms of genotype. And at the end of your differentiation process, you need to be sure that the cells that you have at the end of the differentiation process is from the same identity. We also have to evaluate the potency in order to be sure that the mechanism of action of these cells is stable, and also safety and stability. Basically, in our context, we generate RPE banks a couple of years ago, and each year we need to perform all these some of this quality control to ensure that during this time of conservation, the cells remains the same during the conservation time. You have an example for our embryonic stem cell line, which is a clinical grade. Basically, this cell line was derived by a company named Roslin Cells in Edinburgh, in Scotland. And basically, we identified its morphology through microscopy, its genomic stability through a classical karyotype, its functionality through ability to form embryoid bodies. Basically, embryoid bodies are composed of the three germ layers, and with this kind of assay, you can be sure that you have pluripotent cells able to give rise to any cell type of the adult body. You characterize their phenotype also by FACS for pluripotency markers, and also other assays. And now I will go more into detail into how we generate RP cells from human embryonic stem cells, and then you will see how we generate the tissue engineered product, which is the end product to be implanted into patients. So first of all, how we generate RP cells. The generation of RP cells is quite easy compared to other cell types because they are pigmented, so they are easy to distinguish during the differentiation process. And basically, it's a process which is easy but long because it takes, as you can see, more than 100 days to generate the RP cell bank. So basically, we take some vials from the human embryonic stem cell bank in order to amplify the cells. So we have 3 weeks of amplification. Here you have a characteristic structure of embryonic stem cells, of pluripotent stem cells in general. They form a small colonies of cells that you can see here. And after confluency, we start the differentiation. So the differentiation duration is quite long, but basically, in order to maintain the pluripotency, we use FGF2. And when we start the differentiation, we switch the media and we remove the FGF2. And after a couple of weeks, you can see in the dish the apparition of pigmented clusters. And we collect manually these pigmented clusters, and we allow them to amplify into new dishes. And after a couple of weeks, we have this characteristic epithelial structure, which is characteristic of RPE cells. And at the end of the process, of the clinical process, we generate vials of 200 tubes, 200 cryovials, banks of 200 cryovials. Then when we have this bank, we take a couple of vials from these banks and we characterize the cells upon culture. And basically, RPE cells are epithelial cells, which are characterized by a couple of epithelial markers specific to this kind of cells. So we have the apical expression of ezrin, the nuclear expression of Paxis, as you can see here in green. We have also a specific marker apical of MRTK. We have also basal markers like bestrophin and so on. So we have a population. We also characterize these banks. We need to be sure that we do not have residual embryonic stem cells into our final RP cell banks, because these remaining embryonic stem cells could generate teratomes when grafted into animal models or humans. So we need to be sure that we do not have residual cells. So when you take the embryonic stem cell line before differentiation, you can use a combination of LIN28, which is a marker of pluripotency, and TIRP1, which is a marker of RP population. And basically, before the differentiation, you have more than 90% of embryonic stem cells. And after the differentiation process, you can see that you have a majority of RPE cells. So with this kind of assay, you can be sure that you do not have residual embryonic stem cells during the differentiation process. Then we have some functional assays to demonstrate that these RPE cells are functional, meaning that they can exert native RPE functions. One of these functions are the ability to phagocytose debris of photoreceptors who are generated during the process of light detection. And basically, you expose RPE cells with debris of photoreceptors which are labeled with FITC, and basically you see whether how the phagocytosis occur with this kind of assay. And basically, we demonstrated that the RPE cells that we obtain are able to phagocyte photoreceptor debris. I put it here, the quality controls that we used in the clinical bank. Basically, these quality controls are required to use these cells for a clinical application. So, as I told you at the beginning, you have to characterize the identity, the purity, potency, safety, and integrity of the cryovial. I'm not going to enter into details, but you have to specify the method. Some methods are according to the European pharmacopée, in particular assay regarding sterility, endotoxins, and viral safety. And then once we have these RPE cells, we had to think how to implant them into first animal models to demonstrate the proof of concept, and then into patients. So there are two strategies that were proposed. The first one is to take directly the cryovial and to tow the cell and inject it directly into the eye of patients. And the other strategy was to generate a tissue. As I told you, inside the eye, the RPE cells are organized as an epithelium. So therefore, we are not sure that if we inject directly the cells as a cell suspension, they will be able to integrate within the RPE endogenous layer. And moreover, in a diseased eye, like RP patients or AMD patients, maybe, we are not sure whether the cell suspension will survive. It was written here, and basically the two strategies are tested in clinical trial, and the limitation with the tissue engineering is that you need to design a specific strategy to use scaffolds in order to differentiate it into in order to generate the RP epithelium before grafting. And you need also a specific surgical tools in order to implant it into patient. Whereas with the cell suspension, I would say simply with a needle, you can inject it into the supraretinal space of the eye of patients. So to give you an overview of what's going on with clinical trials. So as you can see, there are several clinical trials all over the world, mainly in the US, UK, Israel, and Japan. And also we have our clinical trial in France. And some of these clinical trials are based on RP cell suspension. The first one in particular were based with RP cell suspensions. And some of them are based with an RP epithelium organized over a specific scaffold, which can be synthetic or not. And all these clinical trials were based on embryonic stem cells, and a few ones were based on induced pluripotent stem cells. So as I told you, the global idea was that an RPE cell suspension would have a reduced graft survival and would not be able to preserve photoreceptors, which are represented here in green. At the opposite, if you organize the cells as a tissue, you may expect an optimal graft survival and functionality. So in our lab, we tested this hypothesis to generate an RPE epithelium. We used human amniotic membranes who are generated at St. Louis Hospital, who are collected at St. Louis Hospital in Paris. And basically, they make banks of small pieces of human amniotic membrane. And basically, we use these small pieces of amniotic membranes that are fixed in this kind of insert, and then we seed over it RPE cells. After a couple of weeks of cultures, you can see that cells start to pigment and to organize as an RPE epithelium. So I'm going to show you how it looks like. So basically, we use the same RPE markers, and you can see that you have a polarization of hydrin in this monolayer of epithelial cells. You have also expression of ZIDO1 at the junctions. When you go deeper into the analysis with electron microscopy, you can see that you have this characteristic monolayer of RPE cells with the presence of microvilli in the apical side, the presence of melanosome, and also here you have the amniotic membranes with the structures of collagen, and here you have the RPE cells. And also in terms of functionality, we performed phagocytosis assay. You can see in this 3D reconstruction an RPE epithelium with the MRTK apical marker, and you have here photoreceptor outer segments in green, and you can see that some of these photoreceptor outer segments were internalized inside this RPE epithelium, as you can see here. Then we observed that in vitro, our RPE epithelium derived from embryonic stem cells correspond to a native epithelium, but we needed to demonstrate that this kind of epithelium, when implanted into an animal model of retinitis pigmentosa, we would expect a visual preservation. So we took a rat model of RP. This rat model have a photoreceptor degeneration represented here in blue, which occur early in life. After the first month of life, you have a drastic decrease in photoreceptor number. And this decrease in photoreceptor number is associated with a visual impairment. And basically, this photoreceptor degeneration is caused by a RPE dysfunction, a genetic RPE dysfunction. And the idea is that if we graft our RPE epithelium into the eye of these animals, we could expect, if it's functional, a delay in photoreceptor degeneration and preservation of vision. So in order to graft it into this rodent model, we needed to develop specific surgical approaches in order to deliver it into this really small eye of rats. And in order to do that, we needed to rigidify this structure, as a biological tissue is really difficult to manipulate. So we embedded it into a gelatin biomaterial, is what you can see here. Then we loaded it into an injection cannula. And then once placed into this injection cannula, I will move tack. Up, you will see that it's folded inside this injection cannula. And then when you inject it into the eye, you need a really small incision. And once it goes through this cannula, when it exits this cannula, you will see that with the gelatin, it opens, as you can see here with this demonstration into a culture dish. And basically, the idea is to make an incision at the end of, at the bottom of the eye, to create a retinal detachment, to detach the retina from the RPE layer, and then to deliver the cell therapy. Once we grafted these animals, we needed to perform some functional assays in order to demonstrate this functionality, and one of the classical assay is the electroretinography. And basically, you expose the anesthetized rat to a flash stimulus. Then you record, depending in with various intensities. Then you record with electrodes the signal elicited inside the eye, which correspond to a B wave amplitude, which indicates the functionality, the ability of the eye to convert the light signal into electrical signal. And using this approach, we observe that we had a preservation in blue. In red, here, in blue, you have the control animal, and in red, you have the animal transplanted with the RP epithelium. We compared this RP epithelium with here, what you have, this bar here correspond to the cell suspension. So we compared the same cells injected as a suspension or as an epithelium, and we observed that we had a better functionality with a tissue organization upon transplantation. We used other tools. Here, it's a heat map showing you the retina. And this heat map, the more it's red, the less you have photoreceptors, and the more it's green, the more you have photoreceptors, according to the thickness of the retina at this different position. And here you have the location of the optic nerve, showing you that you are always at the same position into these different animals that were transplanted with a sham injection. So it means that without cells, with the cells organized as a tissue, RPE cells organized as a tissue or as a cell suspension. And you see that you see a lot of green in the location of the transplantation, of the tissue transplantation, compared to other conditions where you have really a small preservation of the retinal, the photoreceptor inside the retina. We have also made some histological analysis. I'm not going to enter too much into the details, but you have here also the cells injected as a tissue. You can see that here it's a photoreceptor layer, which is the thickness is more important compared to the sham and the cell suspension injections, which indicates that with the injection of RP organism as an epithelium, we have a better preservation of the photoreceptor layer inside the retina, which correlate with the functionality. And finally, I just wanted to show you a last behavioral assay, which is quite simple and intuitive, and show you that how the light signal is integrated into the brain. Basically, you take the rat, you put it into a platform in which there are 4 screens in all directions, 4 screens, and these 4 screens display different bar, black and white, of different sizes, and they are moving in one direction or in the other direction. And with this kind of stimuli, when the rat detects the signal, he has what we call an optokinetic reflex, and he moves the head to follow the movement of these different bands. And with this kind of approach, by modulating the size of the different bars, black and white, you can determine visual acuity. And when we compare the different conditions after 3 months post-surgery, as you can see, we have also preservation of the visual acuity compared. Here you have the wild type, and here you have the dystrophic condition, and here the sham. And as you can see, the animals had the preservation of visual acuity in this behavioral test. And basically, just to conclude with this demonstration, as I showed you, when we organized, we formulate the cell therapy as a tissue, we observed a better preservation of the retinal structure compared to cell suspension. And even if it's more complicated to implement this strategy in clinical trials and in preclinical assays, this strategy is enough provide enough benefits to you to try to develop to pursue the development in clinical trials. When what I showed you for the transplantation into rodents, we had to include the tissue into a gelatin layer, and this gelatin layer provided enough rigidity to fold the RPE into this cannula to inject it into rodent. But this process is really complicated and is really depend on the experimentator, on the operator, and it's really complicated to transfer it to a pharmaceutical establishment to make it reproducible. So we needed to develop a specific medical device in order to make it more simple, without the use of too much technology in order to generate these 2 layers of gelatin. So we developed a really simple medical device that could be applied in a pharmaceutical establishment, who allow to control in red here you have the patch, and in yellow you have the gelatin layers. And basically we have a specific medical device with several caps where we can inject with a simple pipette a volume of gelatin which will fill this specific area in the cap of this medical device. You have here it's not this one. Wait. You have here an illustration of this medical device, how it looks like. So it's really simple. And you have some images in the real life, how the operators work with this kind of device into a GMP facility, into a pharmaceutical establishment. Basically, you have your culture of cells in this specific inserts, where you have the gelatin and inside the RPE epithelium. So you take your culture insert, you put it in this medical device, you put the first cap, the cap 1, you add the gelatin layers in both sides. So on top, on the cap, in this area here, and at the bottom in the other area. You allow it to polymerize in order to have the rigidity. Then you remove the cap. You put another cap used for the transport, and we had this specific packaging to send it to the hospital. Once prepared as this, we call it a therapeutic unit. And this therapeutic unit, there is a delay between the time it is prepared to the time it is implanted into patients. And we needed to evaluate how long it will remain stable before the implantation. And so we tested different preservation period at 4 degrees. Why at 4 degrees? Because we need to keep the gelatin polymerized before implantation. And then it's more simple for the transport at 4 degrees. At a controlled temperature. And so we performed some classical tunnel assays in order to evaluate the stability. And to make it short, we determined that the stability of this therapeutic unit is stable only for 48 hours. So the surgery needs to be performed 48 hours after the preparation, the final preparation. And then we needed another device to simplify the action of the surgeon. So as you saw in the first movie for implantation into rats, we cut manually the patch to be implanted. But in order to treat a patient, we need a reproducible size of the patch. So we designed this kind of buzzer, which is quite simple. It's a kind of a punch with this size, this specific shape and size. And basically, once the surgeon is obtain the therapeutic unit, it just have to press into this button in order to release the patch to be implanted into patients. So then we had another challenge, is to develop a surgical approach who would be compatible with the human eye, which for which the structure is a little bit different to the rodent. And basically, I will go fast through this strategy. We use another strategy taking that approach in order to deliver the RP patch into between the retina and the RP layer. Then we had to validate this surgical approach into non-human primates, and we needed to validate all the logistical strategy, from the production in the pharmaceutical establishment to the delivery to the surgery room, where the Dr. Stephen Bertin made the surgeries. And first we did it with several primates in order to ensure that the surgery was safe. And we had several assays to evaluate the functionality of the retina after implantation. So I will not present how it is implanted. It's a little bit long, but following implantation, you have here the fundus of primate implanted with the patch. You see here in white clearly the patch in these images, and you see that the surgeon was able to localize clearly the patch, easily the patch at the macular area here, just under the fovea. You have here a magnification, and we observe that the patch before implantation and after implantation, the patch remains stable during the follow-up of primates. We observed that during this kind of surgery, we observed a transitory inflammation, as you can see here, of the anterior chamber. But after 1 week, it completely disappeared. Then we operated, we did not have access to a primate with a specific retinal degeneration. So in this assay, a primate were wild type, so without retinal degeneration. And the idea was to be sure that following the transplantation, following the surgery and the implantation of the patch, there was no alteration of the retinal functionality. And basically, as you can see here, there was no major alteration of the retinal functionality at the baseline and post-surgeries. Then, at the end of the follow-up, we evaluated the survival. So it was 7 weeks. After 7 weeks, we evaluated the survival of the patch using human RP markers by histology. So you have a primate retina. And here in red, you see the collagen 4 marker, which is a human marker that is specific to the human amniotic membrane, so the scaffold of our RPE implant. And with MRTK, a marker which is specific of RPE, you can see that over this red, you have I have a magnification, yes. You have this epithelial marker in green, which correspond to human cells. And human cells were viable on top of following implantation on top of the human amniotic membrane. You have other images here of photoreceptors in green. You have primate photoreceptors, and in red you have STEM1 121, which is a specific marker which labels specifically human cells. And you can see you have your monolayer, which is in tight interaction with photoreceptors, indicating that the implant was able to integrate into rodent. You have other images into this primate, sorry. You have also other images of specific photoreceptors, markers, EUR G.E.N.E or rhodopsin markers. And here you have also other markers of cone, and basically there was no major alteration of photoreceptors upon implantation. So we were happy of that. We demonstrated that we were able to generate a clinical grade RP epithelium. We developed different strategies in order to have a process which is clinically compatible and suitable for the clinical practice. These devices were easy to use by the surgeon, validated by the surgeon, and used in preclinical studies into into non-human primate. And basically, I did not present you, but all the regulatory safety assay in vivo were completed and without hurdles. Then we completed all the experiments required for clinical trials. So last year we started the first clinical trial, our first clinical trial based on embryonic stem cells derived RPE in patients with retinitis pigmentosa. And specifically, we focused on mutations associated to RPE defect, mainly LRAT, MRTK, and RPE65. In this clinical trial, we assess first the safety and as a secondary objective, we evaluate if we can measure some signs of efficacy. We had treated 2 patients which are almost blind. It was the 2 first patients to evaluate that there is no major safety issues related to all the process, including the implantation and the cell therapy product. And we have just started to treat the first patient of the second cohort. And in this patient, we could expect a visible photoreceptor ONL, so we could expect some signs of functionality. And finally, just to conclude, we are trying to move forward with this first proof of concept that is ongoing in human with this kind of therapy. We are thinking that we are not targeting only RP patients. We are also targeting with this kind of cell therapy AMD patients, age-related macular degenerations. And for this kind of patients, there are many people, millions of patients that need to be treated. So we need to think to industrialize the process. It will have the advantage to reduce the variability due to human manipulation. If we can go through an automation of all the processes, then we will produce large scale RPE banks with homogeneous population and reproducibility. And of course, we will reduce the risk of human contaminations, so associated with classical cell culture. And in particular, the more we generate doses from one batch of production, the more we will be able to reduce the cost of each treatment. So at iStem, we have this kind of robots who is able to perform some automations of culture steps. So with this kind of system, we are able to culture human embryonic stem cells or human-induced pluripotent stem cells. We are able also to develop protocols to differentiate these cells into RPE cells. It's what I'm going to show you here. So we have designed a specific protocol in which we needed to adapt several parameters. In particular, the first protocol that I showed you to generate RPE cells is based on a manual collection of pigmented foci that appear during the culture differentiation. It's a spontaneous differentiation protocol, but to have an automation of this differentiation process, we need to guide the differentiation, to force the differentiation into RPE cells, to not have to collect only RPE cells among the whole culture of cells. So we used specific cytokines and small molecules in order to differentiate these cells, and we were able to bypass all the manual steps associated with RPE differentiation, and we were able to generate banks of RPE cells, large banks of RPE cells. Now we are switching to another kind of robot, which is compatible with a pharmaceutical production, but it will be the same strategy using a similar protocol. So just to finish, I would like to thank all people involved in this project. As you may understand, this project involved many people I do not have the time to sit all these people, but mainly at ISTEM with Christelle Monville and all the other peoples. At the Institut des Biothérapies, where we have all the regulatory part for the preclinical development, the clinical development, and regulatory affairs. We have also a strong collaboration with the Institut La Vision, with Olivier Gouraud, with Stéphane Bertin, who was the surgeon in non-human primate, who was also the surgeon, who is the surgeon for the clinical trial. We have also the companies involved in the design of the different medical devices, Mersenne for the CE Mersenne for the experiment with primates, Mohamed Jaraya at the Saint-Louis Hospital for providing us the human amniotic membranes, and also EFS, Atlantic Bio-GMP, for the pharmaceutical projection, and many other people. And also all our funding from ANR, AFM Téléthon, Fondation de la Recherche Médicale, Fondation Maladie Rare, and Revive. Thank you for your attention.
Well, thank you so much for this talk, and we have received quite some questions in the chat, so I will be reading them to you. So first of all, Emmanuel Cazot asks, what is the efficiency of your differentiation protocol?
Protocol to generate RPE cells. So basically, I can have two answers to this question. First of all, with the spontaneous protocol, where we differentiate embryonic stem cells into RPE cells, and this protocol who was used in a clinical application, in this clinical trial, basically, we do not care of the efficiency of the differentiation, because we can see RPE cells, RPE foci that appear during the culture, and we collect these cells. So we do not need to have an efficiency who is 100%. Because once we have purified these small clusters, then we have a purity near to 100% after this manual selection. For the protocol, automatized protocol, we have almost 100% of purity, of efficiency of differentiation. And all the contaminant cells are removed during the culture steps. So at the end, we have 100%.
Thank you. So Cassandre asks, is the differentiation into RPE cells as efficient with IPS than with embryonic stem cells?
The differences between embryonic stem cells and induced pluripotent stem cells. So basically, there are many subtle differences, but for a cell therapy application, what is really important is that your pluripotent stem cell is able to differentiate in the cell type of interest with a high efficiency and purity. At the end, at least at the end of the differentiation process, you need to have as much as possible a pure population. And you need, when you use induced pluripotent stem cells, you need to have other checks to be sure that there is no integration of the reprogramming factors, no derivation in terms of genetic alterations, and so on. And basically, why all people use embryonic stem cells is that because we did not have enough knowledge about induced pluripotent stem cells to safely use into patients. But nowadays, there were 2 clinical trials, at least 3, who were started with induced pluripotent stem cells, and so far there was no major problems in Japan, and there was one started also in US. And with this knowledge, we believe that we could have a safe use of IPS. And moreover, IPS are more interesting because the use of human embryonic stem cells is different. There is a local regulation who can limit in one country or in another country the use of these cells. And when you develop a cell therapy, you expect to treat as much as patient as possible with the same product. And therefore, that's why there is a large switch to induced pluripotent stem cells.
Thank you. So there is another question from Felix. Is there differences between embryo stem cells and induced stem cell other than their origin? And do the induced stem cells used in this therapy has to come from the patient or not?
In fact, I answered first to this question, and I did not saw the other question. In terms of efficiency between EPS and embryonic stem cells, in terms of RP differentiation, no, there is no differences, major differences. Basically, we have access to several lines of iPS and embryonic stem cells in the lab at iStem, and we observed that some lines could differentiate easily to RP cells or other cell type, and other cell lines are less suitable for this differentiation. But it depends more on the cell line than on the type of iPS or embryonic stem cells.
So Emmanuel Cazot asks, human female pluripotent cells are known to be epigenetically unstable regarding X chromosome inactivation because of culture conditions. Do you check for the inactivation stability at the beginning of the differentiation? Does it hamper the differentiation, and do you think it could be the source of long-term problems in grafted patients?
I could directly answer to the last part. Do you think it could be the source of long-term problems? First of all, as I told you, we have several lines in the lab from male or females, IPS or ES, and we did not observe major differences regarding the ability to differentiate into RPE cells and in terms of their final functionality, at least in vitro and in preclinical animals in rodents. The most important problem related to the long-term problems in grafted patients, I think it's in terms of survival, maybe the question. The survival is more related to the need for immunosuppression into patients in order to, as you have an HLA which is not matched between the donor and the recipient, the grafted cells and the eye of the patient. And in that context, we need to use an immunosuppression. And we need to mitigate the risk of immunosuppression because with using immunosuppressant, you have several side effects, and you need to have a balance between the level of immunosuppression. And that's why some clinical trials use local immunosuppression, for which we are not sure that it would be efficient, and some use high immunosuppression. But the major problem related with the long-term survival is the use of immunosuppression. We are thinking that it would be a problem to have a long-term immunosuppression in grafted patients, because you will have a lot of side effects. And to limit this problem, we are thinking about controlling the HLA of the grafted cells. Controlling the HLA by matching the HLA of the donors with the recipient using haplotype banks, or through a genetic modification of the donor cells in order to knock down the HLA1. There are several strategies published recently to address this point.
Yeah, thank you. And so Louise asks is kind of what you were touching on just earlier, and is there any risk of rejection of the transplanted tissue?
Would complete that with the fact that we are working with the eye. The eye is supposed to be an immunoprivileged site like the brain, so with limited access to immune cells, but there was a lot of preclinical studies performed in rodent and other animals models, and basically there is a huge discrepancy. Some some studies report that there is an immune rejection when you implant cells into the eye. Some others say no, there is no. In our hand, to be sure that there is no immune rejection, both in preclinical models we used immunosuppressant, and in rodent and non-human primates, and also in patients we use a protocol of systemic immunosuppression.
Thank you. So same thing, you already answered a little bit the question of Julie. So she was asking if there is a risk of developing an autoimmune disease or any chronic inflammation.
What I showed you in rodent is that we in non-human primates, sorry, is that we observe an immune reaction related to the surgery, the kind of surgery with the implantation. There is I did not enter into the detail, but there is a vitrectomy. There is it's a complex surgery using cold liquid and so on, and it's really invasive. That's why we observe this short-term inflammation, but that inflammation disappears, and both in rodents, in rat, in mouse, in non-human primates, we did not observe a chronic inflammation, at least during the follow-up period. And regarding patients, they are treated with an important immunosuppression, but from other studies that were performed, in particular in France, for cardiac diseases, so with the injection of cardiac embryonic stem cells, derived embryonic stem cells, they observed that they looked carefully at the immune response against the graft. It's not autoimmune, but immune response against the graft. And they observed some antibodies directed against the graft, but without clinical signs during the period they analyzed it. But it was really particular in this case with cardiac cells. They treated the patient during a short period for 1 month. And in our case, we have a longer we have a treatment during the period of the clinical trial.
Thank you. And so Jonathan asks, what is the predicted time for this to be available to AMD patients in France? What are the predicted costs? And will it work for both wet AMD and dry AMD?
I will start with the last question. Will it work both with wet and dry AMD? In fact, what we know in our case, the indication was for retinitis pigmentosa, but as I told you, there are some clinical trials all over the world. Some of them you tried to graft an RP epithelium into dry AMD, some others in acute wet AMD. So there is a large variability regarding the indication, but we suppose that in both cases you have an alteration. In wet AMD, you have this neovascularization with an alteration of the Bruch membrane and the RP epithelium. In that case, we would deliver an RP epithelium who would replace the degenerated cells. And in dry AMD, you have the degeneration of the RP without this formation of neovascularization. In that case also, you would expect a recovery by replacing the cells. And in terms of availability to AMD patients, I would say it depends how much money we will have to go as fast as for example, for the vaccine, for the COVID vaccine. The more money you have, the more quick you can go to the clinical trials and then to the patients. And also the predicted cost depends on how much we can industrialize the process. I showed you one step for which we expect to be able to industrialize it, to have an automation compatible with industrialization. It's the RP cell bank, but we are now working in the other step, generating an RP epithelium, and we need to have an RP epithelium that could be available on demand. So meaning you need to treat a patient, you can have access to the RP epithelium instantly, directly, and graft it into patients. So we are seeking to strategies of cryopreservation of all these epithelium to have the dose already available to graft it. So we are thinking to the different step of the manufacturing process, and it depends how much we are able to automatize the step and to produce at a larger scale in order to reduce the cost. So at that time, it's really difficult to estimate it.
Thank you. So if there is no questions anymore, I would like to thank you for coming and for speaking on such an interesting subject, and thank the audience as well for being so, yeah.
Just a last slide. So there is a job opportunity. So we are working in a collaborative project with a physicist, and we are generating a complex layer with photoreceptors and RPE to transplant. And we have a collaboration with this physicist, and we have a 5-month contract starting from March 21. And this contract will be in collaboration with CEDUSEN, Centre de Nanosciences et Nanotechnologie, and our lab at ISTEM. So if you are interested, don't hesitate to contact me.
Thank you. And I will just shortly remind you that this session, the seminar was recorded, so you can find it in a few days. You will be able to find it on the YouTube channel of the EUR G.E.N.E and on the website. And we will I want to let you know that our next seminar is on January the 12th. And we would really like to see you again in 2021. And yeah, don't forget to give to the Téléthon. It's very important. And thank you again, everyone.
4 or 5 of December, so this week. Thank you for your attention, and good luck. Bye.